Very excited to present Mike Kelly from NervGen Pharma. Mike, take it away.
Thank you. And thank you for the invitation to present NervGen. NervGen is a pharmaceutical company based in Vancouver. We've been around for roughly 5 years. We have some pretty interesting technologies. We're public on the TSXV as well as the OTC. So I will be making some forward-looking statements in this talk today. You can find our risk factors on cedar.com. So NervGen has a first-in-class neuroreparative therapy. That's a mouthful. This technology was discovered from Jerry Silver's lab at Case Western. I'll go over that in the next slide. Our lead asset is 5 weeks away from human proof of concept readout. This could be a transformational event, not only for the company, but for the science behind neurorepair and how people look at spinal cord injury. This is a cell-penetrating peptide. It is currently fast-track designated, EMA orphan approved, and we just expanded our expanded access policy. One of the patients in the trial, a physician who's representing that patient, requested additional medication, so we have an ongoing EMA or expanded access policy. Let me talk a little bit about the technology. As I mentioned, Dr. Jerry Silver and his team discovered NVG-291. There were several discoveries that led up to NVG-291. The first, Jerry Silver did his life work studying something different than what other people looked at. Everyone was looking at how to get nerves to grow. And Jerry was looking at, well, why do nerves not grow in certain places? What are the guardrails that are keeping nerves from growing all over the place? He started with the optic nerve. Why does it grow in a straight line? And he discovered that there's a group of molecules, chondroitin sulfate proteoglycans, that act like the guardrails that design and guide nerves to grow where they want to grow. They are ubiquitous throughout your central nervous system. They also cement as you develop those synapses together. So with this discovery, he also discovered that they could also inhibit repair. So they're very good at guiding nerves, but they're also very good at blocking nerves from regenerating and regrowing. So the analogy is, if you cut your hand or if you break your foot, your body repairs itself very well. But if you damage your central nervous system, CSPGs prevent that neuroregeneration. So then he went on a quest to figure out, well, how do I turn these things off? And through a collaboration with Harvard, he discovered the receptor that CSPGs attach to cells and signal into cells. So now that he had the culprit and the receptor, he and his team started designing drugs to interfere with this process. So his team, actually Mark DePaul, who's on his team, who's now with our company, head of research, discovered a group of molecules, and NVG-291 was the most powerful in turning off that signaling. It's a cell-penetrating peptide. So it gets into the cell. It's mimicked after the wedge domain caused by PTB signal with inside the cell, and it disrupts the signaling process. And this is well documented in many academic publications. This is one of the preclinical studies. There's over 30 preclinical studies with this drug in multiple different indications and models. But this graph here on the right side of the screen shows serotonin axons below the level of the injury resprouting. So we have a lot of histological evidence as well as biological evidence of regeneration and remyelination. I have a ton of slides on all of our preclinical data, but since we're 5 weeks away from human proof of concept data, I just want to show you one. If you could advance this slide and show just the left, this is a study done by Dr. Rink. This is a placebo-treated animal with a thoracic 8 compression injury. Obviously, the hind legs are not working. There is still some connectivity. There's some movement in the hind legs, but there's no coordination. After treatment with NVG-291R, which is the rodent version, one amino acid different than the human version, you can see what happens on the other side. Could you show the other video? There's obviously connections being made here, or reconnections being made here. The reason I show you this is if we can get one fraction of this to transfer into humans, this would be a breakthrough in science. So let's talk a little bit about spinal cord injury and who has spinal cord injury. This is an affliction that hits mostly men, 75% mostly men. Average age is 44. The average age is growing with the population slightly. And most of it's caused by a vehicle or a fall. They get hospitalized, and I'll talk to you about their pathway in a second, but the rest of their lives, in an instant, their lives are changed forever. They do get better over the first 6 to 9 months, and then they hit a plateau. And then after that plateau, their life becomes, how do I live with my current limitations? So during the first 9 to 12 months, the early period, there is some spontaneous recovery. And that's why when we did our study and we did the animal models, we have evidence that we work in that first 6 to 9 months. After that plateau, we believe that we're the only company that's ever shown any proof after that plateau that you can actually restart that recovery again. And in animal models, we have data that we can restart that. The key takeaway from this slide is we interviewed a lot of people with spinal cord injury. The media has set the bar very high, and a lot of devices have gotten a lot of accolades for people taking a few steps with multiple devices on their body. These folks just want some of their independence back. Every single answer that they gave us led under the umbrella of independence. They want to do the daily tasks of life that you and I take for granted, that you and I could do in a minute, that take them 15 minutes. So the bar here is extremely low. If we could turn back on, if we could turn off the inhibitor and allow them to work and rehabilitate and continue that and not hit that plateau, that's the goal here. The impact on patients, the healthcare system, is immense. Each patient costs the system between $1 and $6 million over the course of their lifetime. That's a big range because it depends on the average age of the patient's injury and the severity of the injury. When we talk to payers, when I first joined the company, we did market research and I just wanted to get a gauge from payers. Do they know this impact? They know this impact. They know it extremely well. This is a $50+ billion hit to the US healthcare system. I heard a talk, and I still can't find the footnote, about the Canadian healthcare system. This is the #1 cost to the Canadian healthcare system, is spinal cord injury. These folks are rehospitalized with urinary tract infections and other illnesses throughout the course of their lifetime. I said we talk to payers. Let me explain this a little bit. There's roughly 12 million people worldwide living with spinal cord injury, and the reason we take these cuts from this marketplace, there's 350,000 or 300,000 in the United States, and the reason I'm focused on the United States, that's the first market we're entering. So if you take the first cut of that 12 million, you're at 310,000 in the US. The next cut is cervical patients. Although we don't believe the FDA will restrict us to cervical alone, that's the group that we're studying. Our current clinical trial is C7 or higher, and I'll go through the clinical trial design in a minute. We then even took a further cut, which is motor incomplete versus motor complete. So there's 3 different types. There's the cervical, thoracic, and lumbar. We took the cut for cervical, and then we took another cut for motor incomplete. And the importance of motor incomplete when I get to the clinical trial design is there's some residual sensation or function below the level of injury. There is a connection. Most pharmaceuticals, and this, believe me, I've been talking about this product for quite some time. The first question I get is, this is a graveyard. So many products have died coming up to this point. Most drugs, if not all drugs, have studied motor complete injury in the acute setting. meaning you have to get their drug on board within hours to days after the injury, and they're focused on the most severe patient population, motor complete. We're focused on C4 through C7 motor incomplete. So there's still, majority of these folks are still wheelchair bound, but we're looking to enhance the connections that they already have there. It's a big point. And when you apply what the payers said that they would expect us to charge, putting into the orphan space, obviously this depends on the product profile and how much we can give back to the patients and the healthcare system. But if you take the low end of the orphan couple $100,000 range, this is a multi-billion-dollar market opportunity. So let me jump into our clinical studies. This is our phase 1 study. This was done over COVID. It was single and multiple ascending dose. The key takeaway to this slide is this was done in healthy people, healthy volunteers. Everyone asked me, did you see any effects? No, we didn't see any effects because they were healthy volunteers. They weren't paralyzed. But the key takeaway is there were no serious adverse events. The drug was well tolerated. There was one adverse event that was common, and it was common in 90+% of the patient population. That was injection site reaction. So this is a peptide delivered 1 ml subcutaneously every day. And it's not that they were getting rashes or irritation around the injection site every day, but throughout the course of treatment, 7-week treatment, they did have at least one injection site reaction. Now on to our clinical trials. There's a little background here. Our preclinical studies showed we worked in subacute, acute, and chronic. So that's the way we looked at the different patient populations. Obviously, we're trying to make this patient population as homogeneous as we possibly can, and I'll talk to you about some of the entry criteria on the next slide. But the goal here was to focus on motor incomplete. That's the hypothesis from the preclinical trials, and to focus on some biological effects. And so we do have a biomarker that we're using in the study as our primary endpoint, and it's called electrophysiology. So we're actually physically measuring the electric connection going from the brain past the site of injury to a hand muscle group and a leg muscle group, and I'll get into that quite a bit. We also have functional endpoints, which are typical in all of the clinical trials. So this is a 16-week trial, 12 weeks of treatment, 4 weeks of follow-on. And the reason why we're following these folks for 4 extra weeks after treatment is because in the preclinical studies, we saw that the rodents actually continue to improve. So once you remake those connections, the benefit doesn't stop. The connections are made, and it takes a while for muscles to continue to plateau. So we saw some benefit post-treatment period, so that's what we did in this study. This is a single center study. It's done in Chicago at Shirley Ryan Ability Labs, the #1 rehab hospital in the US and potentially the world for the last 30 to 35 years. And the PI on the study is Monica Perez. The reason we chose Monica and Shirley Ryan Ability Labs is because Monica is an expert in electrophysiology, and at that single center with a trained team, we reduced the variability of electrophysiology significantly. The other reason is that it's the #1 rehab hospital. Every single one of these patients comes in 4 to 5 days a week for an hour to exercise the specific muscles that we're looking for connectivity and function on. So this is not just an exercise routine. This is actually a structured rehabilitation program for 16 weeks. So it's a bit of a commitment. Folks think it took us a while to enroll. We enrolled this study in 14 months, 20 patients, 10 on drug, 10 on placebo for the chronic group. The subacute group is currently enrolling, so we expect that to be completed next year. But the chronic group readout is going to be in the next 5 to 6 weeks. The key eligibility criteria that I talked about, they had to have a motor incomplete injury. So there was mins and maxes on the functional capability of these folks. If they were too functionally advanced, they weren't in the study. If they weren't able to initiate a step, even though they couldn't walk, they weren't in the study. But the most important thing was that they had to have an intact MEP amplitude, meaning when Monica sent the signal through stimulation down past the site of injury to a sensor on the hand and the leg, they had to have a registered signal. So it was even a little bit further than, it was a biomarker assessment of motor incompleteness. Why do we choose MEP amplitudes? Because there's a direct correlation. This is one animal study, but there's multiple animal studies and there's several human studies that show that there's a direct correlation with improving MEP amplitudes and improving function. This looks at two different types of serious injuries, and it definitely shows a correlation between when MEP amplitude improves, so does functional improvement. So our hope here is when we get this data is to hit statistical significance in MEP amplitudes and trends, significant trends in the functional endpoints, and approach the FDA. The regulatory experts that we're working with feel that this biomarker, just like any biomarker that approaches this division of the FDA, has a pretty good shot if we can nail both of those endpoints of potentially getting fast track designation. So what is an MEP amplitude? MEP amplitude is a motor evoked, motor evoked potential amplitude. It's actually the same way that an electrician is going to measure electric current in your household. It's a pretty rough way to look at it, but the corticospinal path, your motor cortex sends a signal down your spine, which is in the green line here, and then it connects to a synapse out to your peripheral nerves. And you can see on the hands and on the legs here, there's small little sensors, and you can actually measure the amplitude. If you send a signal, you could see the difference between the peak and the trough of that amplitude. And on the right-hand side of the screen here, you see what happens when you have a damaged central nervous system or damage or spinal cord injury, your amplitude is decreased. So the objective of this study here, and Monica tests these folks at baseline, week 2, week 4, week 8, and week 12 to see if there's progression of improvement of MEP amplitudes in these patients over the course of the study. So she sends a signal down the arm to a registered sensor on the hand, as well as down the leg to a registered sensor on the leg, and you can actually measure the distance in that amplitude. They take 15 measurements, they average them, and they normalize it so that we'll be able to look at individual patients and the population as a whole. All of the secondary endpoints are functional. The bottom two are the traditional ones that we see in every single spinal cord injury study. They're subjective, they're not very accurate. So Dan Michael, who won awards for the design of this study, is our Chief Medical Officer, put some specific tests, mostly for upper extremity function, that are much more sensitive than the upper and lower extremity motor scores. So these are also measured, majority of these are measured at baseline, 2, 4, 8, and 12. So what we expect to see at the end of the study is that the population that has been on placebo has no improvement. So it should be pretty much flat line. This is for the chronic cohort. You got to remember, these folks have plateaued 6 to 9 months. So the entry criteria is 1 to 10 years post-injury. The first 17 patients that Dan presented last year, the average time post-injury was just over 3 years. So these folks have just absolutely plateaued. So we expect to see no improvement in the placebo group and consistent improvement in the treated group. So if we can get an MEP amplitude statistical significance with one of these endpoints, we think we have a home run. We have great patent protection. We have global rights to the asset from Case Western. We've built upon this patent portfolio significantly with method of use applications. We're just starting the formulation patents on top of that. So at a minimum, we get into the late 2030s. And we also have another program that's behind NVG 300. Obviously, with a small startup company, you have to put your money on the lead horse. We've done some studies in NVG 300. We just announced a few months ago that we had positive data in animals with spinal cord injury as well as stroke. So our hope is to get that product teed up through the confirmatory animal model studies, through formulation development, and then start the IND enabling studies. We're targeting right now stroke, but that decision hasn't been finalized. We could take NVG-291 and go into stroke as well. I didn't share any of that data here. It's up on our website. We have some pretty phenomenal data in stroke, especially for early lesion treatment. So the bottom line is we have data coming out in 5 to 6 weeks. It's going to be a transformational event for us and we believe for science. We expect that after that, our cap structure is going to look a little bit different than this. We have roughly 70 million shares outstanding, add in the options and warrants, you're up to in mid-90s. Insider ownership is about 20%, 17 of which is one shareholder. It's a high net worth biotech investor based in Boston. Our market cap has, like small companies in this market, has held pretty solid, as opposed to, we were kind of lucky we were on TSX and not on NASDAQ over the last year and a half, because our market cap has actually grown, doubled in the last 2 years. And we're excited that we have a big news event coming up in the next quarter. I'll stop there to see if you guys have any questions.